1 minute. 1 minute. So, welcome again. And here we are for the last and afternoon session. The chairman will be Baldwin Ode. He's our mentor of bioacoustic and the backbone of Xenocanto. Please.
Thank you, thank you, thank you. Well, that's too much honor, maybe. But I'm really happy to be here. I'm also very happy to be able to introduce some older and some new friends even in this session before coffee break. And of course, you see first this picture and name. This session is especially dedicated to Gianni Pavan. And we're really happy also to have his son here present, so thank you for being here. So I first will ask Cesare Brigio to come. I'm very happy to share some bioacoustic ideas with you already, well, and yes, in future maybe even more.
Yes, thank you. Please. Thank you. I would like to say that I am speaking also on behalf of Filippo, because we both shared the acquaintance of Gianni Pavan. Gianni would have deserved something much, much bigger than my bad English and these few slides can deliver. I ask you to try to grasp our affection for Gianni from the few words that we will sharing with you. As I just stated, Gianni cannot be defined a close friend of ours. We interacted in many situations, and I find astonishing the fact that even in the few opportunities that we had to interact, to join forces, to exchange ideas with Gianni, those small opportunities were sufficient to give us a particular imprint of the view of Gianni on all the living world and of his exceptional capacity of vision and of conceptualization of the ideas. And maybe I can say on my part that it all started around 2010, when I had been collecting recordings for several years, and I was trying to find something that could act like a mentor for me, explain to me what was going on in the bioacoustical field, and Gianni was there. In 2013, I and Gianni, with the collaboration of other people, tried to launch sort of an informal network of Italian bioacousticians that would include not only the scholars, the academical people, but also amateurs like me, people who recorded for their own enjoyment, but that had the intention of giving a scientific character to their investigations. In 2013, Gianni hosted at the University of Pavia the first meeting of that informal network, and A few months later, just 4 or 5 months later in Vicenza, we had another meeting. That time was hosted at the Museum of Natural History in Vicenza to set sort of a point, an overview of what was going on the scientific research in the bioacoustical field. And in 2014, we were credited as collaborators of the CIBRA, that was the Center for Interdisciplinary Bioacoustics founded by Gianni at the University of Pavia, and that was the actual beginning of a more close collaboration also in the terms of sharing recordings, sharing impressions, and we began to get closer and closer. Still in 2018, here in Rovereto, there was the workshop whose title you see on the top of the slide, and was another opportunity to get Gianni, that is world noted for his researches on marine mammals and was also a keen ornithologist, to have him engaged more deeply even in the field of Orthoptera sound and of insects in general. The memory of his father, that was one of the most important myrmecologists in the Italian history, was also very dear to Gianni, that explained us how he enjoyed getting involved in the insects, also in the memory of his father. Between 2010 and 2019, I was undeservingly involved in the paper about the rediscovery of Uromenus annae. And Gianni did not participate as an author, but helped greatly both Filippo and me to improve the clarity of our scientific discourse. My personal defect is prolixity. I use a lot of words, much more than they actually needed. And some observations by Gianni helped me focus on that defect and be a little less dispersive. Another suggestion that we derived from Gianni is to make clearer illustrations with easily readable horizontal and vertical scales. That was another thing that we didn't pay much attention to before talking with him. The top moment of our collaboration with Gianni was in 2019, when I am one of the organizers of Entomodena, the entomological event that takes place in Modena twice a year, in April and September. In that occasion, in April 2019, we devoted an entire morning to a bioacoustical workshop in which Gianni was the keynote speaker, and in which he delivered a very, very far-reaching speech about the importance of ecoacoustics, both in the conservation and in the investigation of scientific the scientific use of bioacoustics. Our main work with Gianni was an extensive paper about the analysis of ultramic recordings. We will say a little more words on ultramic in a few slides, with the analysis of 37 recordings not published before, made with the wideband 0-125 kHz. And this gave the opportunity to redescribe the song of many species whose song was previously only described in the audible band. Gianni was well aware of our attempt to exit the limit of anthropocentrism and to deliver a more complete and appropriate description of the songs by including also those inaudible components, and was very, very keen in the review of the manuscript that, in our humble opinion, was quite well done. Since December 2021, I have been discussing a lot of different ideas about the deployment of a network of sensors, an array deployed in a wide extension of territory that could give a three-dimensional size to the exploration of the audioscapes. And many ideas were emerging and were being discussed. Because Gianni was deeply involved in technology. Since 2009, Gianni and Ivano Pellicella, the man behind Dodotronic, producer of cheap and effective ultrasonic microphones, engaged in discussion, and I do not know all the details, but Gianni was very, very involved in the first generation of the Ultramic microphone, and from 2009 kept constantly in contact with Ivano for its successive improvements. Gianni was only personally, I am still using them when recording on personal computer, Windows-based machine with the USB microphones. The real-time view offered by C-Wave and C-Pro is very, very precious and is controlled by a great deal of user parameters, so it can be optimized to anyone's need. Gianni, summarizing what we know about him, was first and foremost a good listener. Every time we tried to draw Gianni's attention to some topic, he read our mails very carefully and was attentive and responsive, both in personal conversation and in mail exchanged. He declared that it was a principle for him to keep his mind as open as possible, and when talking to somebody, his mind was open in your direction. He was very keen to discuss even the most eccentric proposals that I made, and every discussion with him was very, very productive. Gianni was an eclectic, absolutely. He considered himself as an eclectic. There you see a short email excerpt in which he says, it's unfortunate for us, the eclectics, to be engaged in so many things. But Gianni was what we may refer to as a visionary genius. Gianni seemed to work at an higher level of abstraction than yours, and was able to draw to his level of observation your ideas. And anyway, he could also master the details. He had this capacity of double focus. Gianni was also, but I must say also first and foremost, a teacher. It was impossible not to learn from Gianni. Was eager to explain. He was always available to new engagements and was very capable in involving other people. And was also a developer of software, of concepts, and as I stated before, of visions. My presentation is very, very short, but please, I ask once again, try to imagine all the things that I could not explain and that made Gianni such a very welcome person in any group of people in which he wanted to engage. Thank you.
Thank you, Cesare. Questions seems a bit odd, yes, but maybe there's some additions. Maybe somebody has another story about Gianni to share. Yeah, Paolo, come. But keep it short.
Yes, of course, very short. No, I had the luck to work with Gianni in last years about bees. I involved him in the topic of the song of the queen bees, And we made this research in a very particular place, that is Elba Island, where a very skilled beekeepers teached us how to produce a sound with the voice in order to stop, to freeze the bees in the comb. And so Gianni was very interested to this topic, and we made an excursion of some days with students that made her thesis. And it was nice for me to experience this peculiar topic, that the object of the recordings was Homo sapiens and Apis mellifera too. It's not very easy to do this. And we were working over on this Queen song, because it's strongly debated. We don't know so much of the biology of Apis mellifera as we can suppose. And it was really a tragedy to realize that his life was finished so rapidly. Excuse me.
Thank you, Paolo. So I will ask now David Bennett to come forward. Yes. David Bennett has been speaking on the former ECOC meeting, and he was then starting with automatic recognition of sounds of grasshoppers, and I'm interested how far he got. David, please.
Not quite as far as I'd like, but hopefully far enough. Okay. So as Baldwin just said, today I'll be talking about the passive acoustic monitoring scheme we've been using with audiomorph recorders when compared to traditional transect methods, and the machine learning classifier that we have constructed for them in northern Germany. So we know that in recent years, passive acoustic monitoring has become very important for birds and bats, and it lets us survey them at the landscape level. Now, originally, a lot of these studies were very expensive, like the recording devices would cost hundreds or thousands of euros, and then analyzing the amount of data by hand would be like no amount of encouragement could get our students to help us analyze all of those files. But fortunately, for these species groups, automatic classifiers have become available, and the cost of the technology has reduced quite a bit. So to give you some idea, these are the audiomorph devices. Unlike the mighty bat logger, they only cost 80 euros a piece. And as we'll see, their performance is quite good. So in our current study, we had 3 main objectives. The first was to design, build, and test an automatic classifier for those species that live in Schleswig-Holstein, to compare the performance of the audiomorphs with transect methods that we would normally use, and also with the more expensive 1,000 euro a piece bat logger. Finally, we tested to see if adding additional devices would increase the number of species we detected. So do we need just 1 device per site? And I'd like to put air quotes on site as a bit of a spoiler. Or do we need 2 or 3, and so on. So this work was part of a wider landscape entomological study I did. So using remote sensing and GIS Voodoo, I looked at the quantity and fragmentation of semi-natural habitat across Schleswig-Holstein. And then we deployed 41 sites at wildflower compensation areas across the state. And the idea was, by having so many different landscape contexts with so many different types of surrounding landscape, that hopefully we would get all or most of the species in Schleswig-Holstein. That was a somewhat naive hope, as we will soon discover, but an attempt was made. So at each of these 41 sites, we deployed a single audiomorph. At a subset of 8 of them, we deployed additional units, so 2 extra audiomorphs for a total of 3, plus 2 bat-loggers. The following year, in 2023, at 7 sites, we deployed units again and compared it with transect netting. As I was analyzing this data, I added all the confirmed recordings of species to a CNN classifier, thank you, brain, to build in the open soundscape architecture, to build a automatic classifier. The idea being that future users could use the classifier we've built instead of going through all 5 terabytes and 2.5 million files by hand, which was fun and completely worth the scoliosis it has now given me. So, a bit of a break from my voice, thankfully. What does this actually look and sound like in reality? So this is from my favorite site near Timmendorf. Aha. Okay, so can anyone guess what species we had there? Exactly. And we were overjoyed. Like in this case, I didn't label Rosaliana. You'll see on the next slide. But it highlights both the good and bad of passive acoustic monitoring. Because it was wonderful. In Schleswig-Holstein, Cortypus dorsatus is very uncommon. And we didn't know it was in the neighborhood until we saw it on the audiomorphs. Then later on, we did a dedicated visit, and it was like, oh yes, it's definitely here. The machines weren't lying to us. You probably noticed, however, that Griseoptera was a little bit sharp. And it and Rosalie, at least in Schleswig-Holstein, are both incredibly common and have a psychic sense for where you've put the audiomorph. So they come up, they sit right underneath the microphone, and they sing for hours. I have thousands of recordings which are just the red bar of Rosalie with the occasional, like as it goes into evening, Griseoptera starts like, wonderful. Like, I know you're here, but you're drowning out everything else. And the moral of the story is don't put it on high gain or have your headphones on maximum without looking at the recording. Oh, no. Thank you. Okay, so the labeling process, again, I didn't label Rosalie for this. Using Ravenlight software, I tell the software, okay, that's Griseoptera, that there be dorsatus. And doing that many thousands of times helps us to build a classifier. But to leave the classifier to one side for a second, just to compare the hardware, the audiomorphs and transects had both very good performance, but overall, I'd say audiomorphs were superior to transects, with one key exception. So they detected, audiomorphs got all bar one of the species that transect did. At 4 of our sites, they detected more than the transects. For example, audiomorphs were much better at detecting nocturnal species and Leptophyus punctatissima, which is ultrasonic. So when you're doing a transect, unless you have a bat detector, you can't hear it. Mammaliotetics maculatus was not observed on audiomorphs, but that's my fault, not the audiomorphs. It's because we started this part of the study too late, and by then maculatus had stopped singing. So it was seen visually but not heard. To look at the time and number of units, over the 3-day period that the bat loggers were active, and please note the air quotes of spoiler, there was no difference. They detected the same species. The bat loggers had slightly higher quality, maybe, but not 900 euros of extra quality. However, and that spoiler pays off, the bat logger battery only lasts for 3 days. The audiomorph, even with nice cheap AA batteries from Netto, lasts a lot longer. And because it lasted a lot longer, those Rosalies eventually started to move, and other stuff hopped into position. So by day 14, when we went to change the audiomorph batteries and card, we had on average 2 additional species. Adding additional devices, so 3 audiomorphs instead of just 1, did not lead to improved species detection at those sites, but, which sounds amazing, but there's a couple of big caveats to that. Firstly, this was in the summer of 2022, we did this part, and as Lisa said yesterday, it was the hottest and driest summer northern Germany has had ever. It was nearly 40 degrees, dry as a bone, and my students and I would go out and risk death in the name of science. So these conditions are ideal for the Orthoptera that were present on our sites, for singing, and for a bit of physics to feel fancy. It's also the most conducive for sound to travel. It's also worth noting that these are wildflower areas in Schleswig-Holstein, so not particularly species-rich communities. Very few sites had specialists, and they're quite open, not many obstacles to sound traveling. If it were a more specialized habitat with more interesting species, I believe this conclusion would reverse itself. And it's also worth noting that here we had Rosalie, as you can see, and Conocephalus dorsalis. So this was a unit which was at position 1, same time stamp, 25, no, it was more than that. It was 40 meters away. And you can see that the quality of the Conocephalus dorsalis, it's a lot harder to see. Like it's at the very edge of what's usable, and that was on high gain. So there was a little bit of luck involved in that, I think. So take that, it's an exciting thing, but probably best to err on the side of caution and deploy more units. To look at the classifier, so far we have 17 species, which all have a true positive value of at least 80% and a false negative rate below 10%. So as you can see, it's mostly fairly common species, but we also have some exciting ones like Decticus vericivorus, Cortipus dorsatus and mollis, and we're working on adding more. So that's Stephophyma grossum, which was for a long time my nemesis, and the sound recording for it has not loaded. As said, we're adding more species with dedicated visits, so we go to where we know species are. We carpet bomb the areas with audiomorphs at different distances and different background noise values, and very quickly generate reference libraries of hundreds or thousands of recordings to add to the classifier. We are essentially cheating, and I apologize for nothing. So far, this strategy has worked well for 6 species, and I have plans to add as many more as possible. We're also loaning out audiomorphs, and I must thank Mark Schenziel-Orts for that. He's helped us with Isophia krausi and Gryllus campestris. I also thank the farmers, our students, Klaus Rieder, and everyone who submitted to the online databases, which helped us a lot. Thank you very much for listening.
Thank you, David. We have time for one short question. Anyone?
Did you have reference recordings or reference descriptions of all the species signals already, or you have to describe some?
I had, in every case, reference signals, for some more than others. So back when I first started, some species only had like 5 or 6 reference recordings. Others had a lot more. And I will say, like efforts like Xenocanto, iNaturalist, more and more reference recordings are becoming available every day, which makes it a lot easier. But yeah, like, it's Schleswig-Holstein. There's nothing too exotic or exciting up there.
Still, there is some research things to be done. Thank you, David. Thank you. So we skip to Florent Prunier, which sounds French and is of course also French, but he spends most of his time in Spain doing really nice things with Odonata and Orthoptera.
Thank you, Bodwin. I'm a little ashamed of my presentation since, well, I will not present so many results as an ongoing project to involve more people into Orthoptera. First of all, I just would like to have just a short forward on climate change, because recently I did a field work in rivers in Andalusia, and even common species like Alnus glutinosa, they are in some places they are really dying out in a few years. And also we are seeing that in dragonflies in Northern Europe, in lowlands, the effects are dramatic. So I think that it will be really important in future for Orthoptera to see as those lowland species which have a northern feeling. I will just give you a little impression of different projects I am involved with, just for you to understand why the level of knowledge and why I still need to learn a lot on Orthoptera. So thank you. So actually, I'm a nature guide and also a consultant of in environmental. So I don't spend all my time on Orthoptera. And back in 2016, I was really happy and to join for the Congress, and I talk about the Cazorla mountain range, which is one of our hotspots in Spain. And 2 years later, I expand about adding all the bibliography we have in Spain. So the current situation is that in the 70s, the 80s, there were quite a number of academics, people who did their theses and joined the university. So we have a very good background of information on about the mountains. But now we are in a transition phase where we need to involve more and more amateurs and people which are not professionals. Okay. So to my earth, the most important thing for me now is I moved to a small village of 300 inhabitants in the mountains, and this is where we try to set up a field station, and so we are doing various kind of surveys, and of course we need to come back to the grasshoppers and the Orthoptera. So in Andalusia, in the region where I live nearby, there are above 100 species, and in our region, let's say, it's around 200 species. Yes. So the size of the territory, to give an idea, Andalusia is double size of the Netherlands and half the population. Usually the most interesting Orthoptera fauna is located in the mountain range in the southeast. And in the southwest, the mountains are much lower and there are much less endemism. Okay, so this is our projects which are dear to our earth, and I hope that in some times we had the occasion to meet there. So we try to receive people who want to know about the biodiversity in our region. Okay, so I'm a traitor as a Bodwin state, so I'm also involved in the dragonflies. Good. So the project we are talking about now is we were selected. We made a proposal and we were selected to present a project on training, and we've called it TEOS, and this is about it. So the project is within a call by Tetris. So we are in the training part of the Tetris. Tetris is a project, and is a cluster of different of 70s entities in Europe, and this project was a third-party project, so open to other entities. So our NGO is quite a small NGO, and of course it's well, we are very happy and proud to collaborate. No? So in the project, we are collaborating. We have 3 partners, and Bosque Animado, also here in Italy, World Biodiversity Association, and the Biodiversity Conservation Lab in the University of Ioannina. And there is a part of consultants, the scientific team, which is operating a project to promote recognition of sound automatically. So, so that's the goal of the project is basically getting people fresh to the material, so try to foster the interest for Orthoptera, and especially to train people to record Orthoptera sounds. And especially in Spain, we will really try to focus to get increase the community of people interested. No? So that's really the core of the project, and with the experience they are gaining through the training, we hope that many people are going to the field and record and contribute to Xenocanto, and then the team of naturalists will implement there are machine learning to promote automatic recognition, so that in a second step that can be included in citizen science webpage like observation, for instance. Okay, so we have 4 main events. So this year we will have just right after the Congress a workshop where Baudouin and Filippo will be teaching. And during the summer we will also move to Spain, to Sierra de la Demanda in northern Spain. And the Spanish workshop will be done in Spanish, so that most people in Spain, amateurs, aficionados, very often they are limited. The access of knowledge is limited because we still don't have a wide community of people mastering the language. No? So we will have 4 workshops, so 2 this year and 2 next year. So next year we'll go to Greece and we'll come back to the southern Andalusia. So all this adventure, if we want to call it like that, is linked with citizen science webpage, so that, or for me, I prefer collaborative webpage, so that everybody can participate. So much beyond the scope of professional ecologist or zoologist. So we will rely heavily on Xenoconto and also on observation. So this is the example of an internship which is now collaborating, and we've been already recording some species which were not represented in Xenoconto. So we have about 2/3 of the species of Orthoptera in the Iberian Peninsula, which have sounds, which produce sounds which are meaningful for identification. So this is the target, of course, of the project. And within this slot, yes. So about 69%, a little less than in other Mediterranean Peninsula, are represent. We have recording of the species, no? So really, the Iberian Peninsula is a bit lagging behind. And here it's the data sorted by families. And well, I think the most important point here is to see that the Bradyporinae are the most underrepresented currently in the data set. Of course, there are many species, cryptic species, and not easy to find the field. And still, also nowadays with a smartphone, most people have quite good microphone at hand, or at least to have some basic recordings, no? But still, it's still not a huge habit. And well, during the winter, we will really aim to identify people who can have recording, so to help uploading to Xenoconto if people are willing. Of course, we are also just inviting you to make access to your recordings. And that is nearly all. So this is the project. And see you soon in Andalusia, hopefully. Thank you.
Thank you, Florent. Thank you. Are there any questions about this initiative, which is great, of course, because we need more people to record in the field? Yes.
Thanks for the great presentation. Do you guys have any plans of working with schools, like incorporating this into the curriculum or something?
No, at the moment it's really getting a community of naturalists, no, of people which not necessarily knows about Orthoptera, but which are really, I will say, serious in the sense that they will invest their time in recording. Of course, after people will do what they want with their training, of course. But at the start, it's really aiming at naturalists. Yeah.
Okay, I think. Well, we have room for one more question, but we can proceed.
Okay.
Thank you. So there's a lot of new developments going on, and there's even a person from the other side of the ocean arriving here, Francisco, to talk about software that may help us to analyze Orthoptera sounds. So, please.
Thank you. Hello, everyone. My name is Francisco. I'm from Chile. I am currently in my fourth year, finishing my fourth year in soundscape ecology, PhD in soundscape ecology at Purdue University in the United States. But before that, I was a music teacher. I taught for 7 years, but I've been always interested in the sounds of nature. I started with birds, and I had a project back in Chile for outreach to teach people about the sounds of nature, and I started recording a lot of birds, and I was very into birds. And then I realized I have a lot of sounds of birds, but I don't have insects. So I did a quick query on the Facebook group of the bug nerds in Chile, and I asked them, who is the sound expert on the insects here? Nobody. There are 2 people who know how to identify dead insects, Orthopterans, but nobody knows about their sounds, so you're gonna have to do it on your own. And at the beginning, I was like, oh, I don't wanna do this. Actually, I didn't pay any attention to insect sounds before when I was, recording the birds. And I got a little bit depressed when the spring was over and the birds stopped singing, and I didn't have anything to record, apparently. Until I got this notice that I had to do this work. And I started doing it, and I am obsessed about it now. It's super fun. It's like collecting Pokemons or whatever. I don't know. I'm not into that, but I feel like it's similar. So hopefully more people start doing this. And when I started doing it, I faced a lot of challenges. First, to identify the species, because in Chile, the knowledge about Orthoptera is very little. And to identify the species, we rely on very old descriptions in German or Latin, and we don't have access to the type specimens all the time, because most of them are in the US or in Germany or in France. We want them back. Anyway, so it was very difficult to identify and to link the sound with the species. We're working on that with some experts in Chile. And then the next challenge was analyzing the sounds. So how do we do this? There's several software available for bioacoustics or other that it's not dedicated to bioacoustics, like Adobe Audition, for example, but allows you to make some analysis. But the problem is that some of them, like the pro versions especially, are very expensive. So that's prohibited for most of us in Latin America, for example. And I thought that the solution might be to use free software. So I know that several of you might use R. And there are other libraries connected to R, like Plotly, to generate interactive plots, and Shiny to make web applications that are completely free. So it took me some time to learn how to do these things. And finally, I was able to create an option for the needs that I had. And then talking to Cesare, Filippo, and Gianni, I realized that this might be a good deliverable for the research community, especially to encourage newcomers to bioacoustics to do this thing. Other challenges are like, that we didn't have a standardized nomenclature about the terminology, what is a pulse? That's one of the most controversial terms in bioacoustics, especially in insects. The Q factor etc. And also, are my recordings good enough? Why didn't I use the same equipment in all the recordings? So there's a lot of things that you have to think of. But hopefully, with the development of these tools, it's going to be a little bit less of a burden. Not all of the people are keen to use programming languages, right? So I decided to do this a Shiny app, which is with a user interface, and it has several functionalities, from importing the audio into the R environment to be available for analysis. So you have a multi-page Shiny app, which is like a web application. This is the first page where you can import the audio files into R, and then you have some more pages for pre-processing the data, like cleaning the audio a little bit without modifying the parts that you're going to analyze. Then 2 pages for analysis, and then another one for standardized plotting. So here you just browse, you click on this thing, and then you browse your computer to search for the files, and then you save them, you add them like a custom name. Generally you use the species name. This first page is to downsample, so sometimes you record with an ultrasonic microphone, but the insect call is very low in the audible range, and once you when you're sure that that's the case, it's might be useful to downsample to enhance the computer's performance, and to also facilitate the plotting, because there's a lot of space that you're not going to use to plot it. So what do? You first analyze visually the spectrum, so you see what the maximum frequency of interest here in this case is about 40 kilohertz. Okay, then you find the minimum Nyquist frequency, which is like the effective range of the recording that you need to be analyzed, and that's double the maximum frequency that you are interested in. So if in this case we have the 40 kilohertz maximum frequency or high frequency, you need at least 80 kilohertz of Nyquist frequency. And I limited the options here to keep some consistency in the graphs. So you have 96 kilohertz, 128, and something else, I don't remember. So you choose the next higher Nyquist frequency to resample it, and then you basically clip the recording. And it's much easier to work with afterwards in R, which is not designed to analyze audio. So it can be a little bit slow sometimes when you work with high frequency samples. And then you can apply a high pass filter to get rid of the low frequencies that are not your focus, your target sound. So here we have maybe another species singing, and you have a low pitch noise that is pretty much impossible to avoid when you're doing field recordings, especially near urban areas. So you want to get rid of that. So what you do is you select the file that you already imported, you click on mean spectrum, and you see this plot here. This is not for publishing these plots, it's just for visualizing and navigating these functions. So you can also use the spectrogram view to double check what you saw already in the mean spectrum. So here we see, not very clearly, but here was the sharp peak that we saw in the mean spectrum, and then you can confirm that it is a different sound, it is another species, so you don't need it in this analysis. So then you can select, for example, 4 kilohertz high pass filter, so everything below 4 kilohertz is going to go, and then you're going to have a clearer signal to analyze. And the analysis, this is like the most useful part, because with the proprietary software, you have a lot of options, and different researchers use different parameters sometimes, and it's hard to compare the analysis that are not made with the same, the analysis that are not made with the same parameters. So here you have fewer options that are useful for bioacoustics in insects. and then you select just the recording. You type in the temperature of the which the recording was made. This is the decibel bandwidth threshold, which is like the cutoff threshold to select or to detect the maximum and the minimum frequency here, the bandwidth of the call. Then you have an option to do total bandwidth. That is like basically total bandwidth would be the maximum frequency at which it's cut by the threshold, independently of the values that are in the middle. So I generally use, I uncheck that option, but for some other applications might be useful. I also have another option which is called robust calculation. I didn't put the graph here, but if you click on this, the profile of the mean spectrum is going to be much smoother, and that might change sometimes the peak frequency, because when you have several peaks that are very close to each other, close to the maximum amplitude, that can vary from individual to individual. That can vary from the conditions in the recording, the surrounding vegetation, etc. So if you do the robust calculation, it's going to be much more generalizable, or at least that's the idea. And you can also change the amplitude scale here from decibel scale to linear scale, which doesn't change the detection of these frequency parameters, but it's just a visual difference here. And this show lines is just to plot the lines. And these plots that are here, you can download it as a PNG with the camera there. This is from Plotly. So you click on the camera, you can download it as a still image, or you can save the plot and it's going to be an HTML file that is interactive. So the users can zoom in, zoom out, and explore this plot further. I should have run that before. So those are the parameters. I don't know how much time do I have left. 4. Okay, those are the parameters. You can ask me about those later. But so this on top are the common parameters that everyone use. But also I added a couple of acoustic indices that are most used in soundscape ecology or ecoacoustics, but also can be useful to characterize the spectral quality of the insect songs. And also you have this other analysis page to get the statistics of the temporal aspect of the calls. So you select the file again, and then you can modify the detection threshold. This is a percentage because the signal is normalized to a range of 0 to 1. So 10% is the minimum call amplitude in this case, and then 5% would be the minimum detection threshold. So what does that mean? The minimum detection threshold would be like this. For example, I can detect this, but the detection will be dropped if the maximum of that signal doesn't reach the minimum call amplitude. I need to change some names here because I'm assuming it's a call. It may be a pulse or whatever. So just terms that I need to change, but the analysis will remain the same. So here you have the number of calls, the mean call duration, so you don't have to do all that manually. It takes maybe less than 1 minute, and you have all these statistics ready to use. And these are the parameters, the measurements that are automatic here. Duty cycle, pulse rate, which is very useful for crickets, mean pulse duration, mean gap duration. And I introduced a new index here, which is rhythmic complexity, which intends to characterize the temporal complexity of the song, regardless of the temperature, because you know that songs change in duration based on the temperature of the environment. So this is normalized by the duty cycle to account for that. And finally, there's another page for plotting. So you generally, when you report this in papers, you have to plot the oscillogram or waveform. And this is made very easy in this application. And a couple more things that I'm working on that will be soon available too, is to standardize the spectrogram views, which are often reported as well. But I've seen in so many publications that the sampling, the window length that they use for the spectrograms are the same that they use for the mean spectrum. And we don't need to do that because this is just for you know, graphical representation. There's no analysis made in the spectrogram view. So this is a little bit blurry, and this is much better to my judgment. So I'm working on an automated way to select the parameters for the spectrogram, so people don't have to do that on their own. And also some extra functions to map and get the coordinates of the recordings. You select the audio files, and then you create a metadata file with all the coordinates interactively, which is useful for when you do spatial analysis and so on. And that's it. Thank you.
Thank you. Are there any questions? Because this is really another topic than we discussed in many of the other presentations. So, any questions? No? Well, it's nice to see this is work in progress, and having all these parameters ready in just 1 minute for a recording sounds really nice. Especially the temporal parameters. I'm really keen on using, maybe even.
Oh, and I'm going to be demonstrating how to use this on Monday. And for those of you who are not going to stay, I'm going to record a video tutorial as well.
Ah, there will be a video tutorial about this. Thank you. So we will have a coffee break until 15:30.
Yes, yes, yes.
ready to start.
So, okay, I ask you to take your seat because we have for 6 talks now, 1 hour and a half. So, please welcome Baudouin Ode. You already know him. He will talk about Xenocanto. Please.
Thank you, Filippo. Okay, so I'm a famous guy in Xenocanto, and I do hope you all, guys and girls of course, will also be famous in Xenocanto somewhere in near future. So I'll talk about some recent developments, and well, let's see what happens. First of all, the situation for Xenocanto in Europe is really improving. So you can see all the dots, which are locations with recordings throughout Europe. We have now worldwide 25,000 recordings, some of which, especially mine, may be quite long. And since 1 week, I'll show you later, there's also a possibility to access Xenocanto from the Orthoptera species file website, the new one. And as we expected, and I've seen that also bird people and bat people are adding to the database, so that's really nice to have. But okay, today focus on Europe. We had some special activities last year with some extra funding, and we hope to raise some automatic identification out of this, disclosing a lot of data from Klaus-Gerhard Heller, Siegfried Ingves, Dragan Shobanov, and myself. And there are some more databases we know. Paolo, yours is there too, waiting, but some of those databases needed extra work because we need also the metadata to be disclosed. So the dots will be on the map, and there will be an indication of the recorder that's being used and other things. So I will be talking a bit about meaningful sounds, and Florent also did a bit about this for the Iberian Peninsula. So we have about 1,080 species in Europe, and about 66% produce meaningful sounds. So those are sounds that may be used in the field to locate and identify species. So, of course, some species produce very weak sounds, and they may be very interesting, but they will be hard to hear when you're in the field listening or recording. And especially there's a lot of recordings from the Acrididae and Tetigonidae, about 93% of the species already. So that's really good. Well, there's no meaningful sound in several groups, which are shown there. They are producing sounds, especially in the Pamphagids. There are some noises made, but, well, Bruno may ask me about it later. And if you see a taxonomic overview of the recorded species within the groups, you may see the percentages of these groups in Xenocanto at this moment. Some smaller genera are really missing as a genus even, so we have to look at those. But of course there's also an overview of the species that may be missing, and I hope we will be sharing a database soon to well, get people involved in filling the gaps. So a geographic view already shown by Flora, with 82% of the species already present in non-Mediterranean Europe, and well, there are some major, or at least a little bit bigger gaps in Italy and the Iberian Peninsula. In the Balkan, there's already quite a good coverage of species, thanks to Dragan Shobanov, I think, but he did a lot of work there. But also we did. So I just to introduce you to Xenocanto, because I heard some people really don't know the website. You can search for a species. The Xenocanto website now contains not just birds, and it also says now sharing wildlife sounds from around the world, and not just birds, because that was the old logo there. You can choose for looking only at the grasshoppers, Orthoptera, and you can search a species. I chose a species which was described by Galvagni, which we celebrated yesterday, more or less. We, Paolo, Roy, and me were visiting Sardinia, and found this species and recorded it, of course. And you can see the distribution pattern is there as well. The small dots are there, so you can see where the species has been recorded. You can see maybe if it occurs on Corsica as well, you could add a recording, because that may be a missing recording of the same species. And you see, of course, a list of recordings below, which you can choose one by one. If you choose one, you may see an oscillogram, a bit rough, not as nice as we would like to have it, and a spectrogram above it. And in this case, it is a recording with both a real-time play possibility, but because it's a high resolution recording, you can also play it 10 times slowed down. I can make you listen to this. So this is the normal version, and this is the slowed down version. And you can hear the pattern quite clearly by listening. So this is really nice. As I told you, it's not just the recordings, it's also relevant metadata, like the microphone that's being used, song types, if it's a calling song or another type of song made by this species. The quality is mentioned. It's not always correct, in my opinion, but we'll see. And in some cases also, and we try to do this as much as possible, the collection number in specific collections is also mentioned and available, searchable, so you can connect to a specimen that's in a museum. So there's an API which physically connects to other sites or apps or databases, whatever is needed. And observation data also go to GBIF. I'm going the wrong way. Sorry. So here is also a representation of Xenocanto in GBIF. Okay. The picture is made with this UV light of a Isovia species. So you see it's really good. But this is a side thing. About the prospects of the website and all the developments that are now going on and may be coming. I think sound recording, it's been mentioned before, has become more easy and cheaper. Also in respect to having several types of qualities available in Xenocanto, it may even be useful to have sound recordings made by your phone without any extra technical help, because these recordings may be produced by citizen scientists and also need to be identified if we have identifiers for the European species. Okay, I told bat people and bird people are joining. There is data validation by peers, but this could be somehow being improved. So I do hope we can work on that. I say we, but I'm just advising more or less the Xenocanto people. I'm not one of them. So I hope they will listen to me. There is a publication in preparation that will describe all these newly additions, and we will use the ICN red list, species list, to tell how many recordings we have for every species and etc. Okay, so the training aspect Florent has already told about. Sound recognition models for grasshoppers are, well, they are now nearly being made. The first version is expected in 2025. It will be an open access algorithm, and depending on how good recordings are, but the practice is that with at least 5 recordings of a species, we would be able to have an identification tool for about 59% of the European species that make meaningful sounds. Okay, again, I go back. I know there's a lot of complaints about the new website of OSF. We might have had a workshop on this website because there is a lot more possible, but it's not clearly available up front. But at least they have made a tab for Xenocanto recordings, as they also made one for our naturalist pictures. So you can easily, also on your phone, choose a species in OSF and play the song, which is, of course, very nice to have. Concluding, the public availability of sound-recorded Orthoptera is growing. There may be some options to find, not just in DNA, but also in songs, some song races, or even new species. Who knows? I hope AI will help in future identification. We will be able then to involve more less skilled people. Of course, especially then, we need to think about data validation, and we will always need to be aware to check the AI outcomes of automatic identification. And to reflect on the presentation of Roberto, Battiston, I still prefer real human or animal music. And it's not just grasshoppers that sing, so I hope I can. Grazie.
Thank you, Baldwin. We have time for one very short question, please, just because you stolen some time from him. Any questions? No. Okay. So, thank you, Baldwin. Thank you. And please, again, Cesare Brizio.
Grazie.
About passive acoustic monitoring. Thank you.
Very good. I am speaking on behalf of a group of 4 people. Antonella Di Palma was central because she was the person who deployed the passive acoustic recorders. And this is actually not rocket science if compared to the exceptional work by David Bennett illustrated earlier this afternoon. You will see that the approach is much, much simpler. And this is due to the fact, we shouldn't be shy about that there was no budget at all for this research that was taken forward in full economy. We know the pros and cons of passive acoustic monitoring that can extend our presence in any given territory, but on the bad side, having to do with recordings from a passive acoustic recorder means that we have to deal with suboptimality in many facets, suboptimal distance, suboptimal selectivity, no directional selectivity, and so on. And this creates a series of problems, and to manage those problems, we had to develop our own jargon and to find our own solutions, maybe not particularly brilliant, but again, very economic to put in place at the price of an inordinate amount of work, but we will explain this later. Bruno Massa has been using the passive acoustic monitoring for years in ornithology, but the unexpected discovery a couple of years ago of Acheta pantescus during the expert review of ornithological recordings led us to think that maybe if we deployed our lucky recorder in some place whose Orthoptera population wasn't well studied and set the recorder so that it would take recordings in regular intervals during night time, we may obtain as a result a coverage, a rough estimate of the coverage of different species in the study area. Our only aim was the assessment of Orthoptera diversity. No statistical data were examined, were analyzed. We aimed at obtaining 30-minute recordings, the same duration that intercepted the songs of Acheta pantescus in Pantelleria. And the only review that we performed was by expert listeners, not by technologies such as Open Soundscape or similar. Also because, as you will see, the number of concurrent songs was so high that the training would have been very problematic. What we put in place was a consistent analysis and diagnosis workflow. 2 locations were investigated, The two stations were not far from each other, yet were able to intercept slightly different species, as we will see in the final summary. During 1 month, in July-August 2023, 17 consecutive days in each location. Among the factors of suboptimality, the low quality setting of 24 kHz sampling frequency, passing band up to 12 kHz, was aimed at ensuring the longest possible duration of the storage without storage depletion. The recorder was activated for 30 minutes each hour from 8 p.m. to 5 a.m. The soundscape was heavily contested, meaning that there was much noise and up to 7 or even 8 Orthoptera species singing simultaneously. Our ubiquitous and first in foreground, singing in foreground species was Platyclase intermedia. Many recordings were only long stretches of very loud Platyclase intermedia songs. The issues about suboptimal quality and about lack of selectiveness required sort of a manual activity of decollisioning, deconfliction of the different songs by manual digital filtering. We evolved the concept of surviving acoustic signature with reference to the song that survives a contested audioscape where disturbance, noises, other species do not permit that the full range, the complete frequency range and the detailed distribution of pressure patterns as available, for instance, in a reference audio recording that was obtained under optimal conditions. Well, in this situation, a surviving acoustic signature is what is preserved in the conflict with the unfavorable acoustic environment of the same song. Now, the problem is that we must, by expert evaluation, expert listening, reconcile the ideal situation of the sample obtained in ideal conditions and our signature that survives the confliction with the audioscape. This is a typical situation that we obtained. One interesting area of conflict is between the song of the two Acanthus species, Pellucens and Dulcisonans. And it is very, very interesting that we perhaps observed a Lombard effect. One of the next slides will elucidate this concept in the songs of the two Acanthus. This is a very partial representation of the way in which two songs may collide. Each of these situations requires, if the confliction is possible, to adopt a specific sequence, a specific kind of band stop or band pass filters. Here is the visual impression in the time pressure envelope, in the oscillogram, if you prefer, in the sonogram, call it as you like, between the ideal reference recordings on the left and the respective visualization as an extract from our recordings. We observed that robust characters that survive those kind of conflicts with noise and with heterospecific songs can be defined quite clearly. For example, the two illustrations that you see in the slide currently being displayed show Tetigonia viridissima and the fundamental band of Acanthus pellucens. Robustness is necessary, but it's not a sufficient condition. In some cases, in a IQ song, in a grilled song, where the fundamental and the harmonic bands were intersected by the recorder, the fundamental band was entirely obliterated, and we had only the remaining first harmonic band. The first harmonic band itself was mistaken for the fundamental band of an unknown species, unless we strive to obtain and examine more recordings and understand that we were in a big mistake. This was one of the problems that we have to deal with. What we call as the solution of these kind of challenges is a standard workflow that will be published in a few months in the Atti dell'Accademia Agiata dei Roveretani. You will need patience to obtain the final publication that is quite long, but elucidates our way, our particular workflow in every detail. What we need was consistency. I received something like 350 recordings for around 180 hours of total time, and I needed to devise a way to treat all these recordings in the same consistent way so that I could deliver consistent results. That workflow, I come from the IT sector, as you can imagine, IT and quality control, so my reasoning is very schematic, and this is the way we actually worked. Here are the results. We recognized 19 species, and a couple of these deserve mention. Peronemobius haideni was never reported before for Apulia. Up to our best knowledge, we didn't know that it was observed in Apulia in any time. One interesting thing is Meconema thalassinum. You know probably that with Meconema we are entering the domain of biotremology, considering that that species drums the substrate with his feet. It was very present. As we were not present during the recordings, we do not know whether it was very near the recorder in the leaves of the tree to which the recorder was attached, or if it was at some distance. And this would be interesting because how far the song of Meconema thalassinum travels is, in my opinion, still an open question. The 19 species recognized with their spectral view on their surviving signature, their robust characters in the frequency domain are all enclosed in the illustration currently displayed on the screen. And we discovered that, summarizing our result, the method proved to be cost-effective as long as time has no cost. If you give a value to your time, this may not be so effective. Issues and points to consider with the highest attention include the PAM recorder placement, PAM recorder settings. Among the results, this possible Lombard effect. Last but not least, we should not harbor unreasonable expectation about the result of a passive acoustic monitoring campaign, because it is not demonstrated unless, and we didn't do it, unless you make transect and try to capture physically the specimens, it is not demonstrated that you obtain a full coverage of the entire diversity, also considering the period of the year. Probably one or two of the species in the 17 days in the first location were singing, but in the second 17 days had already stopped the singing seasons. About the Lombard effect, well observed in birds, is the situation in which a species sings outside its usual frequency range to overcome noise or disturbance that occupies its native frequency band. We did not do any statistical research, we did not collect specimen, and we were not there when this happened. Yet our impression, that is well documented in the oncoming paper, is that both Acanthus pellucens and Acanthus dulcisonans may display that kind of behavior. This is all that I had to say.
Thank you, Cesare. Thank you. We have time for a very short question. No? Okay. Thank you, Cesare. Thank you again. Now, Carol Wallach and colleagues, they will speak about the endemic, Carpathian endemic Pseudocoltipus tartrae. Please.
Okay, so thank you, Filippo, for the introduction. Before I start my presentation, I would like to say that when I was coming to Rovereto for this conference, I was quite stressed to present my research as a new person to the field of orthopterology, but the past days of the conference and everything has shown me that the whole community is very nice and supportive, and the atmosphere is super nice here on the conference. So big. So I'd like to thank the organizers and all the participants. And now moving to the topic of my presentation. Yes, we'll move to the new mountain range on this conference. So it has been shown already multiple times that the mountain areas harbor many specialized and endemic species within them. And as you can imagine, it's also the case for the Carpathian Mountains range. And to name a few of those species, we can list, for example, the Isophia hartzi, Hortipus smardi, and many more. And one of those species is the Pseudorthipus tartrae, which is found within few mountain ranges in the Western Carpathians. It inhabits the altitudinal range of 600 to 1500 meters. Yes, endemic. It inhabits the grasslands with sparse vegetation and what is important with the bare ground within it. Usually it's found with low population densities, even to some populations of what we have recorded were singles to few specimens, maximum of around 1,000. And it has quite an enigmatic natural history, because since its description we have very poor knowledge about its ecology and some of the even morphological characteristics and the bioacoustics. And if we look at the recent paper published about the phylogeny of the Central European Hortipus and Pseudorthipus, we can see that where is the pointer? Yes. Sorry for this. Here? Okay, thank you. So as you can see, it's this reddish cluster here, nested in between other Hortipus species and being a sister species to the Hortipus apricarius. So the aims of our study were basically to broaden our knowledge about its morphology, bioacoustics, and the habitat preferences. So the fieldwork took part in the 2022 and 23, and it was a part of the bigger project of monitoring the Orthoptera fauna of the Polish Tatra National Park. All the specimens were registered, and if possible, the stridulating males were also recorded. And as for the data analysis for the ecological preferences, basically we have downloaded all the records and our newly found ones from the Orthoptera SK website for the Pseudorthipus tartrae and Hortipus apricarius. We have mapped them and assigned them a radius of 25 meters. And within that area, we were determining with the satellite maps whether inside of it there is present forest, bare ground, or the grassland. And one more important thing is that additionally we're classifying this bare ground as a natural one. So for example, the rocky outcrops or the gravelly shorelines of the rivers, etc. or whether they were anthropogenic ones. So for example, the tourist paths in the high mountains and sites near the skiing routes, etc. And in the model, the presence of Apricarius was used as the absence of the Tatra, and we have used the generalized linear model with the binomial present-absence data. And moving on to the results, we have obtained the first records of the species for the Poland. The first one were found by the Anton Krysztin in the 2020 in the Łysa Polana, very near to the Slovakian border. And our next records were in the Hala Kondratowa, within the just below the Giewont mountain, southwards. And it inhabited very peculiar habitat there, which was this narrow strip of sparse vegetation with disturbed ground by the tourists that sometimes would like to walk not on those nicely selected paths, but outside. And as we were investigating this site, we have found no other specimens inhabiting these adjacent meadow areas. So they are only found in this very narrow strip of vegetation. As for the morphology, we have looked into the general morphology of the pronotum. We can see that this is it has typical bent inwards lateral carina, just like the most other Horthippus species. And as for the morphology of the tegmina, we can see that we managed to obtain the record of the Macropterus female and male, and it's not so visible in the Macropterus female, but in the male we can see that the median field is abruptly enlarging. And as for the bioacoustics, we have also managed to obtain the first, to the best of our knowledge, record of this species, and hopefully it will be hearable now. Yeah. Basically very short verses which superficially resemble the other pseudo-Horthippus species. And as a note, I must add that this species was extremely hard to record. I was walking near its habitat for like 3 hours, trying to record one 3D lating male, because even if I managed to approach it, they were very easily straddled. So yeah, I have done it, great. And as for the ecological preferences, we have found that the Pseudo-Horthippus tatrae exhibits statistically significant preference for the bare ground when we compare it to the sister species of Apricarius. And there is also almost statistically significant preference, smaller preference for the grassland habitats. And from the other things that I must acknowledge is that only single observation of the Pseudo-Horthippus tatrae occurred on the natural bare ground, and it was the rocky outcrops. So all the other records come from the anthropogenically disturbed bare grounds. And the Horthippus apricarius, in contrast to this, was found on the natural bare grounds. And moving to the discussion and closing remarks, so the stridulation is superficially similar to the Pseudo-Horthippus, which might be also the case why it was in the past reassigned to this genus. And when we look at the oscillograms, we can see that it's most similar to the Pseudo-Horthippus parallelus, but it differs mainly by the fact that its range, the amplitude, is of 1 level of magnitude higher in parallelus when we compare it to the Tatra. And the Tatra verses are also like a bit shorter. And for the Montanus, we can see that the sounds can be clearly distinguished from the Tatra. And our data further confirms like the Horthippus and not the Pseudo-Horthippus species affinity with the morphological data. So this abruptly enlarging median field, but also the general morphology of the pronotum carina. And one last remark about these ecological preferences. So this species is very highly associated with the anthropogenically disturbed grounds, not the natural ones. So it really raises the question, how did the natural habitats look like before the coming of the humans? when there was no paths in the mountains, no clear cuts for storing the logging timber or the skiing routes. It was our hypothesis that it maybe was associated with the megafauna, which was creating heterogeneous mosaic habitats within the open areas. And we can also speculate that due to the fact how specific are those habitats which it lives in, it might explain also the low population densities observed in the natural conditions. And there is also this interesting case of the Horthippus intermedius, which is inhabiting Central and Eastern Asia, but the historical records say that it was also found in the Western Ukraine. So it's, we are speculating that maybe it's not the same species as the Pseudorthipus tatrae, but like closely related one, given the ecological preferences of the intermedius described in the literature, and also its external morphology. When we look, for example, at this picture with the nicely preserved stegmina, we can see that there also is this pattern of enlarged median field. And in general, the overall morphology also suits the tatrae. So this is a matter for further studies to resolve those relationships. And that will be it. Thank you very much.
Thank you, Carol. Thank you. We have time for questions. Axel, please.
Yeah, first of all, thank you for the presentation. It's very good to see some data on this little known species, because it's really something which has been completely missed in the last decades, I think. And one thing I wanted to mention, I mean, you mentioned the question, where did it live before the arrival of humans, more or less. But we should not underestimate the impact of humans also in the past. I mean, during the Ice Age, the humans were already there, right? And they used fire, and of course there was a large megafauna, as you said. And I think if we think in Europe about many species where they would occur without humans or without the current human land use, we will find a lot of species which actually cannot live without human land use anymore. If you look at the Mediterranean, there's so many species which are adapted to grazing systems, which probably were in the former times even older than ice ages, created by megafauna, but I think the humans had already a long impact.
Yes, I agree. I'm not discouraging the impact of humans on those, and of course many other species. It's just like to get a glimpse, maybe a food for a thought. What was the initially important? What purpose was served by the megafauna, which was creating these heterogeneous habitats, which after the humans have killed the majority of it, we have like only replaced it with the pasture and grazing of sheep, cattle, and so on, which is only like the substitution for this, and it's just a glimpse for how the natural correlations with like the other animals maybe looked like with those insects and mammals. And yeah, but thank you for the insight.
Any other question?
Yes. Why you, in the tree you showed, this species is the only Pseudocortipus within all the other considered Cortipus. Why you still consider it a Pseudocortipus?
I do not consider it. It's just that it hasn't been like officially reassigned to the Horthippus level in like the scientific literature. So officially it's still Pseudorthipus, but we have lots of evidence that it is, and the further work should be done to like reassign it to the Horthippus level. And similar is with the Horthippus smardai, which would be the pseudo. So yeah.
Thank you very much.
Thank you.
So, thank you, Carol. And please welcome Gergely Zoveni and Mark Laszlo talking about grasshoppers in Hungary. Please.
Where is the pointer?
Oh, wait.
just.
Okay. You want a pointer?
Oh, okay.
Is
Maybe.
that one? And
Okay.
okay.
To push. Okay. Dear colleagues, now I'd like to show you the new attempts in the conservation of grasshoppers living in Hungary, the large-banded grasshopper. First, we should see the situation with its habitats. Mountain meadows are of secondary human origin, which were established for hay production and grazing very long ago. And after this long-lasting traditional management resulted in peculiar high diversity and characteristic species composition in that nice areas. But in the last century, especially in the last decades, especially in Europe, an essential change have been done in the management. In Hungary, most of these grasslands were abandoned, and the rest of them, the management became much intensive. Why is it so important for us? Because this leads to a very strong decline in the fauna and flora of these habitats due the habitat loss and the degradations. And in our case, it is much more important because in Hungary, this kind of habitats are the exclusive habitat type for Orthoptera fusca. The Orthoptera fusca is otherwise a widely distributed species in Europe, especially in the mountains of central and southern part of the country. And in Hungary, we have only rather peripheral or somehow relic populations in the inner band of Carpathians in the northern east part of the mostly flat country. So in the last century, Orthoptera fusca were detected from 6 mountain regions in Hungary, and unfortunately, nowadays we can find it only in half of them. According a recent thorough research, we found that of more than formerly known about 30 population, only 8 natural population left. It is very sad, and the situation is especially sad if we see the populations in depth. We carried out a population estimations in each Hungarian populations, and we found that even the largest ones has less than 1,000 individuals, and some of them holds only a few tens of them. The situation especially bad in 2 of these 3 mountain regions, in Bergeny and Bükk mountains, where only 1 and 2 population survived. This situation is very, very, it calls for an urgent and active conservation action. What can we do here? First, First, we can try to save the existing populations by large banded grasshopper friendly management, which means some extensive management by moving, removing of litter, because from our previous works we already know that the species is dependent on bare ground plots with low and sparse vegetations for the oviposition. And it is a possibility to enlarge the habitats and connect them theoretically, because as I told already, in Smolenice in Hungary, the places where are officially forested ones are very complicated to change into any other form of coverage. So there is another remaining possibility to establish new populations in suitable habitats. And let's see what we did do. This project started some 6 years ago in the places where the species has only very, very few populations, as I told, in Bergeny Mountains and Bück Mountains, in cooperation with that National Park Directorates, we choose some places for this project. In Bück Mountains, we choose 2 formerly abandoned habitats where the management restarted in last years. And similarly, in Bergeny Mountains, we choose 3 similar habitats. In a part of them, Orthoptera fusca was known, but long decades ago. So what we did, we translocated individuals in small groups with about a sex ratio 1:1 from the large neighboring populations in the same mountain range into suitable habitats. And after in the consecutive years, we revisited the places to check the established populations. First, let's see the Bück Mountains places where only 2 Orthoptera fusca locality left. This is these are the 2 places where translocation have been done. First is Kismező, which is a very rich Dolina meadow. It is not known as former locality of the species. And the other one is Nagymező, which is a huge mountain meadow mosaic where the species was known, but the last data was from 40 years ago. And in both places, the management were restarted in last decade. So we start the translocations in Kismező in 2018, and in Nagymező just in last year. In Kismező, we translocated individuals in 2 consecutive years, and after a local catastrophe, I will show you soon, another couple of specimens. As you can see, in Nagymező, this year we couldn't detect the species, which may mean that the translocation was not successful, but maybe this is not the case. At least the species was below the detection level. But in Kismező, which I show more in details, the situation is different. this is the place just before we release the specimens. We cleaned we prepared the place with cutting and removing the grassland and the litter. And after, we translocated the specimens in 2 patches. And you can see where we could find specimens in the consecutive years with the estimated size of that new populations. Until 2022, the situation was very promising, but unfortunately, it was a summer forest fire there, which fortunately only the perimeter of the habitat destroyed. But after, unfortunately, the forestry used the dirt road crossing the habitat to depositing timber. And, which is worse, they widened the road only by 1 meter. But this was that 1 meter. There in the road verge, it was a nice steep, small slope with bare plots, the main ovipositing sites of the species there. So it is not surprising that the population dropped after, but no, still there exists. In Persian Mountains, 3 plots were chosen, and the translocation seems to be more or less successful up to now. Maybe the best place is near rate, a former location of the species, where we released specimens in 2021 and 2022, and you can see that the population inhabits a rough half of the meadow, with a good number of population, nearly 90 individuals. So it is the question whether were the translocation successful. In Kismező, we think that yes, but you saw that problem we hopefully solved. In Nagymező, we don't know yet, but after now in Bükk Mountains, we already have 3 populations instead of 2. And in Bergeny Mountains, near it, the translocation seems to be really successful. In Nagyhideghegy also, and in Foltán Kereszt, we will see. So in Bergeny Mountains, we already have 3 or maybe 4 populations instead of 1. If we can speak about any conclusions, maybe we can see that the translocation could be a right tool for this species conservation, for establishment new populations, and appropriate habitat quality on the recipient place should be important for the success. And it seems that the number of translocated individuals have an important role. Maybe there is a threshold, and that additional translocations may foster the successful establishment of the new populations in that habitats. And at the end, I have to thank for my colleagues and students the hard work on the field, and thank you for your attention.
Thank you very much, Gergely. We have time for one question, but I would like to say that I really liked this presentation because I agree on most of your aspects, especially when you say that further reintroductions or further translocations can reinforce the assessment of the population, or can foster, as you say, the success of the new population. This is what we experienced with Zoneriana.
I have a question.
Yes.
In this particular case, I don't know if the Orthoptera fusca of Hungary has a different genetic pattern of other populations, but it could be possible, and I ask to everyone here, could be possible, could have a sense to use specimens from stronger, not so far populations, instead to remove 50 specimens from an estimated population of 800 or 700.
Yes, I agree, but this is which I have.
No,
What I have.
no,
We don't have more.
You consider this
As you saw.
was some reason for which you didn't?
Yes, but we should consider the possible effect of isolation between the much bigger Carpathian populations. We don't have any genetic data. We already collected the samples, and hopefully the analysis will start soon. But as I know, we don't have data of these species at all. It is very interesting patchy distribution over Europe and in Asia, and especially in Europe, the population should be at least in the last tens of thousands of years somehow isolated. There must be, I think, some difference between.
Please, Axel, be fast.
I would think about implementing grazing there, which will also help to get some open spots there, and it might be easier to manage it rather than mowing.
Oh, in Bug Plateau, it is now prohibited because this karstic plateau provide the water for a big city nearby.
Okay.
It's there always.
Thank you. Thank you, Gergely. We have to go forward. Thank you. With Simeon Borisov and colleagues about biogeographic questions in the Aegean archipelago. Please.
Thank you. So after we heard these 2 days, we enjoyed several presentations about mountainous habitats and species. Now we are going slightly down closer to the sea level. Biogeographic questions of the Aegean. I will talk more about the questions rather than give any answers for now, because it's early days of the project. But the Aegean is called by many authors the natural laboratory for evolutionary studies, and it's because it's a very complex archipelago mainland system that consists of more than 7,000 small and big islands scattered with also many mountains. Some of them are very high, and the model species that I'm going to talk about is Pecillimon cretensis, a member of the genus Pecillimon, which is on the other hand, it's the with more than 150 species, it's the most diverse bush cricket genus in Europe and in Eurasia, probably. So I have shown the distribution from the IUCN assessment. It is common on the island of Crete, but it was also found on several islands north and south to the island of Gavdos. And we decided to address one of the questions that remained unresolved in this taxon. What is the origin and the phylogenetic relations of the populations from the smaller islands? So what is their relationship with the populations on the island of Crete? So we, several years ago, my team studied the Pecillimon ionicus group, which has representatives on the west and east of the Aegean Sea. And Pecillimon cretensis is part of this group. Due to monophily, we decided to propose unification of the group systematically so that it includes all the monophyletic taxa. During that study, only very few populations of Pecillimon cretensis were sampled genetically, and it was obvious that there are at least two clades that are very divergent, but it was not the main focus of the study. And also to mention some geological accepted fact that Crete was recently separated at least to two or even more islands, paleo islands, during the Pleistocene. So this is probably the explanation for this pattern. However, the other hypothesis is that human-aided dispersal is the reason for that distribution on the smaller islands. So our sampling, we included material collected from Dragan Chobanov and me during the 2018-2019 field seasons. Material from the collections of the Natural Museum of the Museum of Natural History on Crete, and last but definitely not least, the collection of Jos Tillmans, who kindly provided material he collected during the recent years. So what is interesting here is that we have 26 samples and we have 26 unique haplotypes so far. This is compared to other Pecillimon species and other species in general. It's of course, it depends on the molecular marker. This is the ND2 molecular marker, but with the same marker and conditions, it's quite impressive to me because we have everything we sample genetically, it's different so far. We reconstructed Bayesian phylogenetic tree, and the two main clades were obtained from Western Crete. We have quite a compact group. I have drawn this red line a little bit, a straight line that is not really correct. It's probably a curve between the islands and mountains, but we have mostly two very deeply divergent clades, one from the western part of the island and the western islands, and one from the east and the eastern adjacent islands. And the estimated age that we from the molecular divergence times estimation is 1.4 million years during the Pleistocene, middle Pleistocene, very early. And but what is even more curious about these results are the tiny islands that we sampled. For instance, Astypalia is very close to the eastern population. So the population on Astypalia is similar to some from the Kataro Plateau in Crete, which in this case, we probably, it is either for a really human dispersal helped by trade, or even the region was the center for development of civilizations for millennia, probably, or this could be also natural dispersal. We cannot tell for sure, but the most interesting results come from the island of Seriphos. I apologize to any Greek colleagues for my pronunciation of the Greek names if they are wrong, but the island of Seriphos is very tiny, and it has about 75 square kilometers area. And on that island we found the two clades, actually, the two mitochondrial clades. And it also, on the island of Milos, we found the western clade, which is several kilometers by sea, distance by sea. And from in geological literature and biogeographical, it's commonly accepted that the Cyclades archipelago, during periods of low sea level, are united. The small islands that we observe now are united in a so-called proto-Cycladia, or a united landmass that was estimated to have area similar to the area of modern Crete. However, it is also known from other sources, plants and geological data, that exactly the island of Seriphos and some of the nearby islets, Sifnos, Anafi, didn't tend to unite with the Proto-Cycladia. So this is probably allopatric isolation for a very long time. And this is, in the terms of conservation, this may mean that first we need to research more in the area. We have to visit the small islands, do not have to neglect them. And we have to compare these populations also morphologically and bioacoustically. Meanwhile, because of the hypothesis of human dispersal and the question that some orthopterans and even other organismal groups are common on the have a very wide distribution in the Aegean. This is part of a phylogenetic tree that data was provided from colleagues that work with Elpholidoptera. Elpholidoptera smirnensis, which is believed to also hypothesize that it is held by human dispersal to have this huge area of distribution area in the Aegean. We can see how the splits are much younger. So this is a huge contrast to what we saw with Bacillimon cretensis. I'm not going to go further detail for this species, but yeah, the difference is obvious from the divergence times. So what's next? Definitely further sampling, because we have more islands to compare, and provide more genetic data, including nuclear markers, so that we can see how can we collect the puzzle and see what really happens in the Cyclades archipelago and the nearby islands with this taxon. How many taxa? Also one of the most important questions, because it can also be very important for conservation to know whether this is one species or it has subspecies. This depends on the further data and the interpretation of species concepts, which can be a bit tricky. And a comparative approach to just, since many animals live in the same area, we think that it's smart to compare their response to the same processes, such as sea level rise, climate change, isolation. So we, in other projects, we are working with different taxa, Orthoptera taxa, and even we're starting to obtain some results with colleagues with Coleoptera. So last, I want to thank the Ferraki Fund, from the Hellenic Orthoptera Society, Zoological Society, who provided for 2024 the financing for this ongoing project, and thank again the Orthoptera species file for previous funding for collecting material in this wonderful region. And thank you all for the attention and this wonderful event.
Thank you, Simeon. I suggest we go straight to the last talk, and we make the questions later in the closing remarks, please. Okay, so please welcome Lara Sophie Day. She will speak about species distribution modeling in Orthopteras. Please.
So, hi everybody. We had many high-quality talks in this conference, so I think we deserve some light entertainment now. So, we are now talking about the species distribution modeling in Orthopterans, and when we are thinking about the theory and the history of it, we know that Humboldt was the founder of the modern ecology, we can say. And he was the first person who was like finding out that every species has something like ecological niche. But a simple way to show ecological niche is like this. So we have these different climate zones, and this is very rough, because we all know it's more like this. So we have our grasshoppers, and they do like specific habitats. So for example, this one is typically colored green. It should be more grayish or brownish. It loves sun, it loves the desert, high temperatures, but it doesn't need big trees or vegetation or rivers or something. And in the ecological niche modeling, there's a quite simple theory behind it, or how you can explain it. It's a band diagram. The band diagram shows here all of this is a geographical space which can be inhibited by a specific species. And we have these abiotic factors. You all know that, like sun exposure, precipitation, temperature. But we also have these biotic factors. And these biotic factors are like other species, vegetation, and so on. And the third thing we have is a mobility area or movement area. And this area shows like the distribution range where the species, or the range where the species can go. So how much spreading is possible. And we have like some overlapping areas. So G1 is the area combining the appropriate abiotic and biotic factors. Then, easily, G2, actual ecological niche. But we have some problems because in ecological niche modeling, or also in science, we are never able to get all the variables together. And specifically in ENMs, we do have a lot of problems with biotic factors, because if you want to have the biotic factors of each species distribution point, you need to go to the habitat and collect all the data. So for simple models, it's quite a lot of work. And that's why we are working with G3, the potential and environmental space we are able to model. And this means we are like putting all our knowledge on the abiotic factors, for example, from the World CLIM data set. Here we have 19 variables which we can use. And then we use also the movement area to build our models. And the cool thing about ENMs is that it has a built-in time machine. So we can also model potential scenarios in the future and in the past. And in theory, that looks like this. So we do have our map. We do know where the species is distributed. Then we have some abiotic factors in this part, so variables. And we plot these points on all of these variables. All these variables are also shown here in this 3D space as these gray points. And the red points are the extrapolated, or the environmental points within this big cloud of environment, which is inhibited by the species. And when we're extrapolating this to the 2D space or the geographical space, we get maps like this, so fancy, colorful maps. And I think that's enough for the theory. Let's get into some examples. One of my latest projects is about Meconema. Meconema has two species, Thalassinum and Meridionale. I guess most of you know this species already, have found them, saw them in collections, and so on. Just talking too fast. All right. So, wow, what a service. Oh, thank you so much. Okay, so, yeah, we already have that. So, what we see here is the original or native distribution of both species. So, this pinkish part is the native distribution of Meridionale. The blue one is Thalassinum, and the violet one is the overlapping area. So far, so good. But if we are looking at the recent distribution, man, there is a huge overlapping area from Thalassinum and Meridionale. And just look at that bastard. It has no wings. So how did it came to Europe, to Central Europe? Well, that's quite simple. It's like a blind passenger going through the Alps on cars and trains. And there's already one study which says that they were driving 150 kilometers per hour and the insect was still sticking to the window. So I'm pretty interested how much speed it can get. So if somebody is interested in some experiments on the German Autobahn, tell me. So, but now a bit to the ecological niche modeling part here. So this is a current projection for Meconema meridionale. We see, so the light colors show regions which are not that suitable, and the darker colors where we have some suitable areas. And you easily see that it's a huge space. And when we look at Thalassinum, we have a similar picture. So Meridionale has also some more regions up there in the north, but simply this region would be potentially suitable for Thalassinum. At the end, we do not know if it could be everywhere because it's just a model, but let's see. And if we are looking here at the overlap of both species, we see this gray part is a suitable area for both species. We have this green areas, which is for Meridionale, and the blue ones is for Thalassinum in this case. And I told you that we have this built-in time machine, so we used it and we drove to the future. This is for 2100, and what you see is pretty interesting because here, in this region, which was suitable for Meridionale, or is a native range of Meridionale, it will not be climatically suitable in the future for them. But, well, there's some more space. That's quite good, but is it really that good? We don't know, so we think the next stop is Asia. And I could talk for some more hours about this topic. There's so much stuff to say about that, but I think we should go on and talk about another project. And this is a project about Brüdermella tuberculata. I think some of you already saw this part of presentation when I presented. I still like it, that's why I also include it. And Brüdermella tuberculata is maybe not known by everyone. But it's a quite nice species. It prefers steppe and semi-steppe habitats, open river benches, and so on. And it has a huge distribution range. That's more than 7,500 kilometers spanning from Western Germany up to Eastern Russia. But, well, there are also some red points and blue points. And the red points show the extinct species distributions, which we were able to collect based on material from collections, from literature, and you see there's just some populations left here in Bavaria and some here in Sweden. And we were interested, well, what's the reason for that extinction? And we thought, yeah, well, maybe it's climate change, so it's not that suitable anymore. And that's why we did some modeling. And in this one, we used just the points from just the recent points where the species is still occurring. And we did our model, and we used the Mahalanobis distance. Mahalanobis distance is simply just the distance from a point to the ecological optimum. That means a smaller Mahalanobis distance means more suitability. And that's what we got. This is, in red we have the extinct, and in blue the extant occurrences for Mahalanobis distance per occurrence. And what we see is that they are overlapping. So that means that the ecological space, or no, other way around, the distribution points, which are extinct here, they do have climatically optical, what?
Optical or optimal?
Optimal. Yeah, it's quite nice. So climate is fine. Yeah. And if we have a look at the map, so a lot of colors, we see here this yellow part. These are climatically good habitats. And if we focus a bit more on Europe, we see here in white the extinct occurrences, black are the extant, this way. And we see that everything is quite yellowish. And if we see look at the some habitats, this is from Sweden, the Öland population, maybe, and this is from Russia. And there must be something. That's why we went a bit further into the occurrences. And this one is the Upper Isar Valley, or riverbed, in Germany in 2009. And this one is 2020. So there's a lot of vegetation growing, and this shrinks the habitat for the species. So what that means is that the habitat is shrinking because of the not regulated or the regulated flooding of the Isar. And another thing is, I went to Denmark and I looked at the former collection sites. Yes, we have some agricultural areas and fully overgrown heath, so not suitable anymore. But is there potential recolonization possible? Probably. So climatically, the areas are still very suitable, but the habitat structure has to be renewed or, yeah, I didn't write that on my hand, sorry. Restorated, that was it. Okay, so, computer? Okay. So, the last project I want to show you. Do I have some time, or are you just standing?
1 minute, 1 minute. No questions, 1 minute.
Do you want to hear about the last project, or shall I go on?
No, go forward. No questions, go forward.
Okay, we can do that. So, the last project I want to show you is from my PhD thesis, which I ended last year. So, you see here some individuals or species from the Oedipodines which occur in the Americas, and some here which are occurring in the Eurasian African parts. And they do look quite similar, I guess. So, there was this question. There are two possibilities. Are the tribes more related between the continents, or are the species within one continent are more related than the members of the same tribe of another continent? And we did here quite some work. This is a genomic tree based on hybridization capture data of more than 3,000 loci. And what we see here is a clear clustering. Everything from this blue bar upside is from the Americas. And all from here down to here is from the Eurasian African part. So that means that the species within one continent are more related to each other than the morphologically similar pairs. And we did also some geometrical morphometrics, and this is Sphingonotus nebulosus from Iran, and this is Thanotropus latifasciata from California. And this is just the PCA of all the variables we use, and what you see is no clear clustering. So yes, they are a bit different, I see that too, but if they are not spent, they seem pretty similar, and if you do not have any label with a distribution under it in the collection, you're fucked because you don't know where it belongs. And this is shown in this PCA, that these are really similar. And then we also were interested in the ecological niche. So we calculated the ecological niche for Trimorotropis, which is here in the western US part, and we extrapolated that to Eurasia, Africa. And we have some suitable areas here in southwest Asia. And other way around, we modeled for Sphingonotus, which has some potentially suitable areas here in southwest Asia, and we extrapolated them, and they have some extrapolated areas here in the western US. So that's quite interesting. So they share a same, or no, not similar, like they share not the same, but a similar niche. They do share a similar geometrical space, but they do differ in the genetic space. So, yeah, summing up, you see there are a lot of variables we have to focus on when we are doing ecological niche modeling or assessing other studies. So, a lot to do, but at the end, we should also talk about, do we have some pros and cons? Because 1 minute. Okay, it's the last slide. So we have some pros, yes. Many applications, as you saw, and you already saw that in the whole conference. So many applications of FDM. And we can show the ecological space in the geographical model, what's nice because it's 2D and we can more explain what's going on there. And the usage of these tools just require a basic knowledge of bioinformatics for the simple models. You should still think about all the variables and so on, but yes, the results could be also used as hints for real ecological studies, so like with real field work and not just taking some points from the internet. And it could be also helpful to improve conservation actions. And the cons, it's just a model, so you should always think about how much you rely on this model and how much you trust it. In my opinion, it's a hint and a feature or some editing for a nice study, but I would never present a study with a potential ecological niche and I say, this is the real ecological niche, nobody can say anything against that. So, okay. So at the end, I want to thank, and I think you all agree with me, to Filippo for organizing this huge conference. It was amazing. Applause. No time. And thank you for your attention.
Thank you. Thank you. Thank you, Lara. Thank you. I think we are arrived at the end. We have to do some closing remarks. And while I'm calling here, please, we have 6 seats. So I would like to call Axel, please. And then Luke, Baudouin, Bruno, Paolo, and I would like to call also Roy, but there are plenty of Dutch people, so I would call Josip here, please, for the closing. You can choose. You can choose the seat. And
Test.
While they are thinking about the closing remarks, because they will do it, I will tell you that at 6:30, we will have a toast in a bar, in a terrace here, the Depero bar, but just follow me, I will bring you there. And 6:30. For tomorrow morning, please be in time. We are leaving at 9:15 from parking lot Leoni. You have the map in the booklet of abstract. I will collect the lunch box for you and I will give you before to leave in the bus, because there are, and I am happy for this, there are many of you that are vegetarian, so there are dedicated box for them. Bring your bottle. We will not give you water because we are avoiding plastic, and we cannot give you glass bottles. So please bring your bottle. We will come back from the excursion at around 6 o'clock in the evening. And for those who want the certificate of attendance, please write me a mail and I will send it to you. I have now the results of the poll about the next edition. But just to have an idea, because I'm sure that we have only half of you have voted. Okay? Only half. But most people voted for Croatia, and with the 37%. And and 30% voted for Greece. So, and well, some more than 10%, so I say 4 people voted for Spain.
Looks like a little bit on a Soviet Union press conference. Okay. Yeah. First of all, thank you, Felipe, for organizing it. It was really, really amazing. I think we had really a wonderful program, very diverse, very attractive, and really high quality presentations. I was really very, very surprised about the quality. It was really good. And also we will have a nice excursion tomorrow, so we are not yet at the end of this conference, so we're looking forward to this. So thank you again for stepping forward during the last Congress, and it was a little bit similar like what you did this time with with the voting, yeah, because I think you won it, yeah, during the evening, yeah, exactly. So that was a little bit, and he said, okay, I will do it, and we were all surprised because it was a little bit a joke, yes, because we know it's difficult to organize a congress if you don't have good support. And I know Josip told me that his buildings, the buildings of his university, were destroyed in an earthquake. So it might not be a good option to go to Croatia.
Maybe we find another city.
Yeah, maybe if you find something else, okay. But I also had some talks to other people, and Oliver said maybe in 3 years it could be possible. But we need to find somebody, so we need some discussions on that also after the Congress. And I don't know. Well, there were some votes for Spain. What about Spain? Joaquin? That's possible? I think that would be great, because that is really, Spain was always a little bit underrepresented on the congresses, and moving to there would probably attract some people from that region. That would really be good. And I think maybe in the biological station Doñana you are, right? Maybe it's really a good...
Then we can take the next one.
Yeah?
We have new
Yeah?
building.
You think it's possible? So I have not voted yet, so I give my vote to Spain. because my smartphone doesn't allow me to open the link, actually. Anyway, I hand over to Luc.
I have nothing to add, I think.
Okay, nothing to add from there.
I must say, I'm not thankful for you that you voted for Croatia, because we have a lot of problems. So yes, maybe we are not ready to host the Congress this time, but definitely next one, I think we will be ready. And I need to start answering emails as well. Thank you very much, Filippo, for inviting us and for giving me this chairmanship today. It was my first time to do such a thing.
I don't know what to say. I think I made another few friends, or few, maybe a bunch of friends today, the last few days, and well, I think tomorrow will be great to join with at least part of you. How many will come tomorrow? Oh, that's quite a big. Nice. I will enjoy. Thank you.
Okay, I must say that this was the most terrible Congress for me. It was really awful, because normally in the Congress I attend only to 5–10% of the presentation. And I must say that I attended to all the presentation. I cannot believe in myself, and I will hate Filippo and all of you for all the short rest of my life. But I must say also that I really enjoyed any of your wonderful presentation. I am very, I'm working mostly on bees nowadays, but orthopterans are my eternal love. No, a little lower. And in my stomach. A little lower. So, but to realize that so many young people with very good masters are doing such kind of progress in Orthoptera research is really great for me. And so I want to thank you all for coming here. And I'm sure that tomorrow we'll have fun together, and so give the words to the
thank you. I appreciated very much the presentation of yesterday and today, and I think that the general culture on conservation of Orthoptera is growing very much in Europe, in Europe, not in all Europe. I would like to thank very much Filippo for all this organization. Okay, Filippo.
But thank you. No, I at the end I have to thank all the administrations that made contacts with you, Rosanna Leitch, my head in chief, Jonathan Stanker. But we should especially thank a dark presence in this room that is in the glass box over there, Marco Nave. He solved all these issues and all the problems. So thank you, Marco, and thank you to the direction, the president of the society, Fondazione, and the director. I don't remember the name of the director of the museum. She's not present. But okay. Okay. Anyway. Yes. Marco will suggest me the name. Okay. Yes. I will be 50 in November. This is the first symptom of my 50 years. I think we will meet for the toast at 6:30. We will meet again tomorrow in the field. So remember to answer the questions to the last two, Simeon and Lara Sophie Day, for the last presentation. Thank you, and thank you for coming. Thank you. Thank you. Thank you to the director, Alessandra Cattoi, the director of the museum. Thank you. She helped very much.